Our vision is that essentially everything that future human generations will need for food, medicines, energy will be produced by harnessing the power of enzyme catalysis from nature. Within the CarbaZymes project, we were focusing on carbon-carbon bond forming processes for sustainable industrial needs.
In nature, vital biochemical reactions are catalyzed by enzymes. Enzymes can also be harnessed to perform reactions useful for the industrial production of valuable chemicals, pharmaceuticals, agrochemicals, and other substances much needed in our society. Enzymes work under conditions that are much milder than those of conventional industrial processes, require less energy, consume less material, and produce less waste.
So we use this nature technology, let's say, to create new carbon-carbon bonds and then create new products, innovating product production, innovating product extraction, innovating new medicines, in new pharmaceuticals, in new agrochemicals.
The application of enzymes and related biological systems is therefore considered a key enabling technology for future chemical production that is safe, efficient, and sustainable. This has the potential to greatly improve the quality of human life and to protect the future of our planet.
CarbaZymes has really enabled us to develop new products that take our 3DM platform to a new level, because these products are much closer to the needs of our users.
Enzymes, however, often present limitations under industrial constraints for synthetic applications. However, in our CarbaZymes project, we were able to show how to overcome such barriers. CarbaZymes was funded by the European Horizon 2020 research program for developing novel enzyme-catalyzed processes for connecting carbon-carbon bonds to build complex molecules.
So we believe that this process can be used in the future after optimization for production of these pharmaceuticals.
Enzyme catalysts are the elaborate nanotools of nature, which have evolved to function optimally in living organisms over millions of years. Today, their improvement for industrial applications can be achieved within weeks by further evolution in the test tube, a technology so important that in 2018, Professor Francis Arnold, the pioneer in the field, was awarded the Nobel Prize for Chemistry.
In CarbaZymes, many talented scientists worked together, and this produced a domino effect of discoveries in the field of enzymatic catalysis, and these findings can be applied to make industry more sustainable and greener.
Interestingly, nature has developed many organisms that live in extreme conditions. Some even thrive in boiling water, or Arctic conditions, for example. So their enzymes must be tolerant to harsh and extreme conditions too.
We were successful in adopting new technologies and to demonstrate a novel process for the synthesis of a pharmaceutical intermediate.
In our quest for optimal biocatalysts, we analyzed a gazillion of bioinformatics data accumulated in enzyme sequence and structure databases and produced large enzyme collections that cover a huge enzyme sequence space, which significantly accelerated our research progress. These hundreds to thousands of enzymes were investigated using novel screening technologies to identify and tailor biocatalysts needed to achieve the desired reactions and that are sufficiently stable under the demanding conditions presented by industrial processes.
CarbaZymes gave us the opportunity to significantly broaden the range of applications of our biocatalysts.
And that's exactly what we need from Enzymicals to gain more customers and to broaden up our product portfolio.
Developments in the lab are usually conducted at small scale only, and direct scale up to the multi-ton scale required for industrial applications is generally impossible. A fundamental goal of our consortium was thus to bridge this key gap between academic research and industrial production. We are proud that the efforts and close collaboration within the international CarbaZymes team were highly successful in achieving and even surpassing our challenging and meaningful targets.
I wanted to learn more about protein engineering, and this is the part that we are doing in University Autónoma of Madrid, and that's why I chose to do my PhD in CarbaZymes.
Yeah, I'm doing my PhD in CarbaZymes because it's a very international team, and also because I can produce some important pharmaceutical compounds.
Imagine this. A chemical catalyst must be synthesized each time again from scratch, with high effort in labor and material investment, not to mention significant negative environmental impact. In contrast, our optimum biocatalysts, once identified, can simply regrow themselves renewably, whenever needed, at low cost and with minimal effort.
We can even use those models to find the conditions at which we will carry out, let's say, the best experiment. So it works. And then we transfer our knowledge to industrial partners and they carry on the work.
Developing robust enzymes to catalyze carbon-carbon bond formation efficiently and with high selectivity will thus have a major impact on the chemical and pharmaceutical industries by replacing non-sustainable chemical processes with eco-friendly enzymatic processes.
Before CarbaZymes, I couldn't provide my customers with some of the enzymes that they needed to produce their products. However, as a direct result of the CarbaZymes project, I can now provide my customers with the enzymes that they require. My customers are amongst the largest pharmaceutical companies in the world. Therefore, the legacy of the CarbaZymes project, from my perspective, will be the availability of tomorrow's environmentally friendly medicines, both quicker and cheaper.
In CarbaZymes, the academic and industrial partners have effectively combined their skills and technologies to pave the way towards a more sustainable future. We believe that in the hands of the scientific and industrial community, our new knowledge and tools will have a significant greening effect on the entire chemical industry.
The CarbaZymes team was very successful in developing new tools, new enzymes, new processes for biocatalytic applications in industry. Once our technology is taken up by others, we believe that this will lead to a greening of the entire chemical industry in Europe.